talk about exploitation, domestication, and diversification of Brassica rapa. But really, we focus on how Brassica crops are diversified during the domestication process. Okay, so everyone here is talking about evolution. But really, we need to talk about this guy and his famous book about evolution. And when you open this book, the first chapter comes to your mind is actually talking about the variation of plants and animals and the domestication. I think that's because Darwin thinks those plant and animals and artificial selection actually are great examples to convince people how selection can influence on evolution. And one of the plants that he has mentioned in this book multiple times is Brassica crops for their great morphological variation. And some people are joking that Brassica are actually dogs in the plant world. So let's get familiar with these crops in Brassica. So the major species within this genus are these 6 species. So people have called this interesting relationship as a triangle of U. So at the tip of this triangle, we have 3 diploid. And the hybrid of each of these 2 give rise to an allo tetrapolyploid of these 3 species. So our group are really interested in the 3 species at the bottom, which is 2 diploid Brassica oleracea, Brassica rapa, and their allopolyploid Brassica napus. So let's get familiar with these things. So at the left panel here, you have all the 1 species Brassica oleracea. You must be familiar with this because if you go to market, you will see a lot of these crops. And on the right panel, here we have all the variations within Brassica rapa. Again, single species. Look at their morphological variation due to domestication. And at the bottom, we have the allopolyploid Brassica napus. So this rap seed is famous for, you must know the canola oil. That's from the oil seed of this guy. So this genus also has experienced amazing things on their genome evolution. So it has experienced several rounds of whole genome duplication. So it has 2 rounds of whole genome duplication shared with other species within Brassicaceae family. And this genus alone has experienced another hexaploidization event, and this is commonly being called as whole genome triplication event. And this kind of genome doubling and diploidization process actually could strongly influence on the evolution and the diversification of this genus and these crops. So when we talk about domestication, the first question comes to our mind will be when, where, and how these things were domesticated. And people trying to ask this question in these Brassica crops use historical literature record, like linguistic evidence, and also try to address this question use some archaeology evidence. And also, recently, people towards use genetic markers to address this question. So here are 2 of the recent results try to solve the phylogenetic, the population genetics of these crops. But we actually get very poor resolution. Generally, the conclusion is we can roughly have 3 groups, as you can see here, demonstrated by different color. And these groups can geographically divide into like Europe and Mid-Asia group, South Asia group, and East Asia group. But there are a lot of confounding results. It's not well resolved. And another question we are interested in is commonly during the domestication process, there is a strong founder effect associated with domestication. But for this diverse group, whether they also experience the same amount of founder effect during their diversification is another interesting question that we are interested in. And within this group, actually, there are several interesting questions that we still don't know. Like, for example, this one is landrace in leafy type Brassica rapa used in Italy, which is a landrace. And some people suggest this is the wild type Brassica rapa, and some other people suggest this is the first domesticated crop in this group. But we still don't know whether that's true. And in India, actually, people use this Brassica rapa for their oil seed. And based on the different seed color, people name them as the brown sason and yellow sason, and people consider them as 2 different subspecies. But is that true? We don't know. And interesting, in East Asia, Chinese people derived Chinese cabbage for the leafy type, And people also arguing about the origin of Chinese cabbage. Some people said it's derived from pak choy, other people said it's from turnip, and some other people said it's a mixture of both. So we don't know the answer. So to address this question, we collected the samples from worldwide and represent of all different Brassica rapa cultivars, and we grow them in the greenhouse, and we extract RNA from the leaf, we collect the transcriptomics data, we sequence them use NGS, and then perform analysis use typical NGS pipelines, and then perform several analyses that could help us to address those questions, like the genetic structure analysis, phylogenetic inference, and also the demographic inference analysis. Okay, so not too much on the method, let's look at the result. So here is our result. At the top, we have a phylogenetic relationship of all the collected sample, and at the bottom, we have population structure analysis. And when we associate these things together, it's like very math, right? But when we compare this result with previous study, actually, as you can see, our result has a way much better resolution. So which actually provide us more insights on the domestication and diversification of these Brassica rapa crops. So let's look at more details. Based on this analysis, as you can see, there are different colors which represent different genetic groups, and we find six major groups. At the basal, we have these rapas from Europe, which is consistent with the previous inference that these things were first domesticated in Europe. And then we have this group, which is the Italian language. So is this one the first domesticated crop in this group? The answer is no, based on this analysis. And also whether it's a wild type, the answer is also no. So that's something we don't know before. And then let's move forward to here. This is the yellow sasan and the brown sasan from India. So are these things really two different subspecies, although they have different color on the oil seed, on their seed? So actually, genetically, we can't distinguish them. And let's move forward. So here's the East Asia group. We have a red group, which is pak choy, and a yellow group, which is Chinese cabbage. So actually, these results show there is a close genetic relationship of Chinese cabbage and choy. So we are very excited about this result, but we want to move one step forward. So we use this method called diffusion approximation of demographic inference, and its short name is DADI. And this analysis actually provides us very powerful analysis that we can distinguish different demographic models and find the optimal model that can well match with our genetic data and provide us some inference about the effective population size and divergence time of these species. So we did a whole bunch of simulation, and let's see the results. So when we put all the things together, here is the demonstration of what we found. So on the y-axis, here is actually the time from present to the past. And here, these different blocks actually represent the population and the population split. The width of these blocks actually represent the effective population size. I have labeled the estimated effective population size of these branches, and also these numbers represent the divergence time. So what we can learn from from this analysis is if you compare these two lineages, the Italian language and the Indian sasan, compared to the East Asia lineage, Chinese cabbage and pak choy, actually you can find this group has a much larger effective population size compared to the other group, which indicates the East Asia group actually didn't experience a very strong founder effect during their diversification process. And when you look at this time estimation, roughly this entire process happened about 3,000 years ago. And more interestingly, as to the origin of Chinese cabbage, as you can see here, is indeed an admixture origin. And about 90% of the genome is actually from pak choy, but there are about 10% of the genome is contributed from European section, like turnip. Yeah. And we also did analysis to compare the genetic diversity of different groups. As you can see from here, actually the Italian language and the Indian sasan show very low genetic diversity, while the East Asia lineage pak choy and Chinese cabbage actually has much higher genetic diversity than these two groups, which also support the founder effect of these two groups are actually different. So why is that? So why is that? So there are some literature record, linguistic record, which can help us understand this process. The first thing is when you look at record, in India and China independently, about 3,000 years ago, there are some records about Brassica rapa. So this is actually consistent with our demographic inference about the divergence time of these things. So which indicates these things were introduced to Asia about 3,000 years ago. And also as to, in terms of the origin of Chinese cabbage, actually the earliest record is about 1,000 years ago. By that time, it's not called Chinese cabbage, it's called niudu. So in Chinese, if I translate that into English, which means beef tribe cabbage, which is, totally makes sense if you see, compare the beef tribe with the leaf of Chinese cabbage, that's the name. And so we have some consistent record, consistency between our demographic inference based on genetic data and with the historical record. And we also find some interesting things happened about at that time period, like 2,000 to 3,000 years ago. So what we found is Alexander the Great, actually his troop has invaded India about that time. And there are other records indicate their troops actually take rapeseed as food during their travel. So these troops and these wars actually potentially would introduce rapa from Europe to South Asia. And at about the same time, Chinese people has established the Silk Road between Europe and China. So there are a lot of trading between the two areas. So this actually could potentially provide constant gene flow from Europe to East Asia, which could explain why we don't see strong founder effect in East Asian lineage while we see strong founder effect in South Asian lineage. So I think this linguistic evidence and the historical record actually well matched with what we found based on genetic data. So let's make some, so here's my conclusion. So first, our analysis actually can provide several insights on the Brassica rapa domestication and their diversification process. And our reconstruction of the demographic history is consistent with historical record. And based on our result, we conclude that the founder effect during domestication may be confounded by multiple rounds of introduction. As I mentioned early in this talk, we actually have a lot of ongoing analysis looking at the genomic signals of selection of these different crops. And we also perform the gene differential expression analysis on these different crops, as well as we also try to address whether the whole genome triplication of Brassica of the genus has some influence on the diversification of these crops, but I just don't have time to talk. I'd like to discuss those things if you, maybe after this. And with that, I would like to thank all the members from our lab, Barker Lab. All these people are great guys, give me a lot of help during my research. And also we have great collaborators working on this Brassica. These are Brassica guys working on Brassica, so I really enjoy this project. And these people with these names with both actually contribute to my analysis to this project. So with that, thank you very much.